Hello, everyone. It's Dr. Muzolan Academy. I'm here with the lecture number 5 of our body fluid series. So, our topic will be the titration of amino acids. You have learned about body fluids, its compartments, osmosis. We go molecular. Today, we ask how does a single amino acid behave when we add acid or base in its environment? Why does it sometimes have a positive charge, sometimes negative, and sometimes have no charge at all? And most importantly, how does this tiny molecule help to keep your blood pH alive, the normal pH that is 7.35 to 7.45? Let's find out the answer of all these questions. Isoelectric point. Let's start with the definition. Amino acids are ampholytes, a fancy word meaning that they can act as both acids and bases. In solution, they exist as zwittrines, from a German Zwitter word that means hybrid. A zwittrine has both the positive and a negative charge on the same molecule, but the net charge is zero. The isoelectric point, the pi, is the specific pH at which the amino acid has no net charge. Why does this matter clinically? Okay. It matters as at isoelectric point, electrophoretic mobility is zero. That's how we separate proteins in the lab through our technique that is serum protein electrophoresis. And again, at the isoelectric point, solubility is minimum. Many proteins are precipitate at their isoelectric point that is exploited, or this actually is exploited in protein purification methods. And furthermore, at isoelectric point, the buffering capacity is minimum. The molecule resists pH changes least at this point. So let's move further with the more definition trade. So let's talk about the acid medium or acidic medium. It means there will be a low pH. The pH is on the lower side, means amino acids gain hydrogen ion. So ultimately, they become cations. The positive net charge is on their structure chart. Next is the alkaline medium, means the high pH. And if pH is on the higher side, then amino acids have lose their hydrogen ions. So they become anions. And then net charge will be negative one. And furthermore, at isoelectric point, zwittrine, there will be no net charge on its structure. In metabolic acidosis, the pH or the blood pH will be on the lower side. And further, in metabolic acidosis, amino acids and proteins behave in such a way that in metabolic acidosis, amino acids and proteins become more positively charged. This alters protein function, enzyme activity, and even drug binding. Furthermore, in metabolic alkalosis, the positive or the opposite, happens. Uh, that's why acid-base disorders are not just about breathing; they affect every single protein in your body. Next to the titration rate. Titration is the controlled addition of an acid or base to determine the concentration of an unknown solution. Analyte is basically the substance being measured. The other one is the titrant, that is the solution of known concentration added slowly. So, what about the ultimate goal? Add titrant until all analyte has reacted. The equivalence point, the volume of titrant used, tells us the concentration of the analyte. So, in amino acid titration, we add HCl, that is an acid, or NaOH, that is a base, and monitor pH changes. This reveals the pKa, or the dissociation constant values of the ionizable groups. Let's walk through a classic experiment. In step 1, start at high pH, that is alkaline medium. All amino groups are deprotonated, means there will be negative charge, right? All carboxyls are deprotonated ultimately, and net charge become more negative, right? There will be a net charge that is negative. And the step 2, now add HCl dropwise. The first protons will neutralize the strongest base, which is the carboxyls of deprotonated nature. They, at certain pH, 50% of the molecules have negative and 50% have, again, the neutral charges. This pH is the pK1, right? Or the pKa or pK1 would be for the carboxyl groups. Now, at step 3, continue adding HCl. You reach the isoelectric point. The net charge will be 0. And at the step 4, add even more HCl. Now protons start binding to the amino group, forming NH3 plus at the pH where 50% are NH or the balanced or have no charges, amino groups, and 50% those are with the positive charge or the NH3 plus. That's the pK2. That is for the amino groups, right? So pK1 is for acidic component and the pK2 is for amino component or the basic component. For simple amino acids like alanine, the pK1 is around 2.3 and the pK2 is 9.7. Let's talk about ampholytes. An ampholyte, that means they can act as either an acid or base. They are amphoteric in nature. So water is the classic example. H2O can donate a proton acting as an acid and accept a proton that can or acting as a base. Amino acids are ampholytes because they contain both a carboxyl group, COO group, that donates hydrogen ion or that providing while providing the hydrogen ion, the medium would be acidic. What about the amino group or the NH2 group that accepts a hydrogen or ultimately medium will be basic or becomes basic. Now let's talk about the clinical thing. The body's buffer system bicarbonate, phosphate or proteins rely on ampholytes. Hemoglobin, for example, buffers carbon dioxide or carbon dioxide derived acid in red blood cells because of its histidine residues, which are ampholytic in nature. Or amphoprotic means having dual properties. Right? So let's move to the titration curve. Strong acid versus weak acid. So we'll look at the two curves, right? Start at a strong acid or the HCl curve that is shown in color red. Starts at very low pH as NaOH is added, pH rises slowly, then at equivalence point, one drop causes a massive pH jump. That is vertical line, as you can see, that is rising on a straight path. So the HCl got the equivalence point at 7, right? Or around 7. So means that become neutral, right? What about the weak acid or its curves? As you can see, the curve starts at higher initial pH and that has a flat buffering region around its pK that is 4.76 here, and the pH resists changes or change. Furthermore, equivalence or equivalence point pH is more than 7, the medium would be basic because the conjugate base or acetate is basic. The jump in this case is less steep. Why does this matter for amino acids? Amino acids have two buffering regions, one around pK1 that is being caused by or being provided by the carboxylic group, and one around pK2 that is being provided by the amino group. That means they can buffer both adding both added acids and added bases, right? This is why proteins are excellent physiological buffers. Now, let's move to the titration curve of the alanine rate. Alanine is a monoamino monocarboxylic amino acid. One has amino group NH2 and one carboxylic group that is COOH. Its titration has burst flat region that is around 2.3, and as you know the pK1 is being provided by the carboxylic group. So the pK1 is 2.3 and the steep rise near isoelectric point that is around 6 for alanine. And what about this second flat region? That is around 9.7. That is the pK2 or the point that's being provided by the amino group, amino buffering. So formula for calculating the isoelectric point of amino acid is, that is pi, or the isoelectric point, is equal to pk1 plus pk2 divided by 2. That is, for alanine, the isoelectric point is, if we talk about the pk1, that is 2.3, and the pk2 is 9.7. So we add both these values and divide it with the 2, right? So 2.3 plus 9.7, that is, and divided by 2, that is 6. So at isoelectric point, solubility is minimal. That's why in multiple myeloma, we use electrophoresis at pH 8.6, not at the isoelectric point, to separate immunoglobulins. If we ran at isoelectric point, everything would precipitate or would get precipitated, right? Now, the buffering action, or the max, that is the max at isoelectric point or minimum at isoelectric point. So this is a high yield concept for the exams and clinical practice, right? Maximum buffering capacity occurs within plus minus 1 pH units of pK1 or the pK2. The minimum buffering capacity occurs at the isoelectric point, that is Pi. Why? At pK1 or the pKa, there is 1:1 ratio for uh, protonated to deprotonated forms. The system is most resistant to pH change. At Pi, or the isoelectric point, the molecule has no net charge and cannot easily accept or donate protons. So, blood pH is maintained at 7.4. The major plasma buffer is bicarbonate. The pKa for bicarbonate is 6.1. But hemoglobin, the pKa, that is 6.8 for histidine residues, and plasma proteins with many amino acids or amino side chains provide additional buffering. If you lose albumin, that occurs in the nephrotic syndrome or liver cirrhosis, your buffering capacity drops. So you become more vulnerable to acidosis. So formula for the titration of glycine. Glycine is one of the simplest of amino acids. Just pH has its side chains, or just H has its side chain. It also follows the same isoelectric point formula, pK1 plus pK2 divided by 2. Why should, or why to show glycine again is just because I need to reinforce the concept. But note, amino acids with ionizable side chains like glutamate or glutamic acid, lysine or histidine has 3 pK1 or pKa values and a different isoelectric point formula. We will cover in the session of protein biochemistry. Thank you very much for your attention. So today we learned the zwittrines, ampholytes, and we learned the next point, that is isoelectric point, is the pH of no net charge, minimum solubility, and minimum buffering capacity. Writer's curves reveal dissociation constant value or values and buffering zones. Buffering is max at dissociation constant point and the minimum at isoelectric point. So We will cover this very topic of titration in a bit crisp way and routine session also. So stay tuned for or stay curious and keep titrating. So we'll see you in the protein chemistry lecture, right? Thank you once again.